Welcome back. Welcome back, everybody. We're in the middle of talking about the LGN and going back to the cortex and the way that the cortex is actually wired up. So we're going to start from, and what we broke with was talking about how the LGN is connected to the visual cortex, and now we're getting into how those visual cortex and those visual cortical cells actually work. So what we're doing now, what this image is trying to get us to think about, is that if we were able to hack into and look at cells that were within the visual cortex, so let's go back a bit here, and just kind of look at those cells and see how they respond, and understand, how do they respond to visual stimuli that are being presented through the retina and passed on through the LGN, we would find that they have receptive fields, representations, that is, of the retina that that would be present in the back of those cells. So we're kind of looking at these different cells, and the first kind of thing to kind of imagine is that we have a monkey here, which is one of the first ways they actually characterize this thing. And the monkey is staring at a visual representation of a stimulus here that's being shown, a bar of light. And then what they're doing is that they're actually recording from cells within the visual cortex at different layers and trying to say, well, how does this particular part of the visual cortex respond, and then how does this particular part of the visual cortex respond, and how does that correspond to the overall visual representation that's being presented. And so this is basically trying to say that, well, within the LGN, meaning that thalamus, right, we have this sort of concentric circle kind of organization that we saw with the receptive fields of the bipolar cells and ganglion cells. So this has basically been passed on from the bipolar cells and then onto the LGN, and then finally onto bipolar ganglion cells, LGN, and now being passed all the way up to the visual cortex. So this is kind of representing the receptive fields of these lateral geniculate cells here, centered around where stimulus is being, the center of it is being on center, and this is off center being depicted, off surround rather, being depicted here. And so what this is trying to depict is that by overlapping multiple receptive fields within the retina, we're able to trigger the activation of multiple on-center areas, enough that we're able to stimulate multiple different LGN cells. So this is a receptive field of one LGN cell. This is a receptive field of another LGN cell. This is a receptive field of another LGN cells. These all feed into the stellate layer in the visual cortex, layer 4C. Those in turn then start to converge on what are known as these simple cortical cells here that are located nearby. So these simple cells, as we describe them, are starting to actually integrate information from having a center surround kind of appearance here to constructing, if you will, a representation of the retina here that's more akin to what we might think of in our own visual experience. In other words, having a bar of light, rather than and that they would have a receptive field that is more like a bar rather than a concentric circle. All right, so these receptive fields of these simple cells are composites of the LGN and stellate receptive fields that are occurring, and they're located near layer 4C. So what's the takeaway here? First is that how those receptive fields get passed on up to the LGN, and then that we're seeing in the cortex a convergence of these different receptive fields. So, all right, let's take a look. We take a microelectrode, we plug it into the back of in the striate cortex, and where we're looking for the visual response here. And so this is a slice of the brain, and then what we're considering is that we're having this bar of light that's being presented here. And so we're looking at it. Our visual field is all of this, and then this bar of light is hitting a portion of our cortex, and we're recording from this side of the brain here. And what we're finding is that the receptive field is being depicted here as this blue area here. And if we're recording from a simple cell, for example, it responds a little differently than what we saw for the retina, where we had these concentric circles. Instead, what we have is that they respond to these bars of light. And so because it's a composite of multiple receptive fields that we had in the LGN. So here's our visual stimulus that's being presented. There's our visual stimulus. This little blue area here is trying to represent this receptive field here. And as we shine a light here, we see that there's a cell, this is kind of the amount of discharges occurring. What's kind of interesting is that as you change the orientation of the light so that you're activating more of the receptive field, you ultimately are able to create it so that you actually have a greater amount of firing here. And so this here is actually hitting more of these sort of on-center parts of the composite of all of these multiple different LGN cells here that make up this particular type of the receptive field of the simple cell. So in English, see these things here? These central circles here represent each of these different types of LGN cells. Concentric circle, concentric circle, concentric circles here. So one LGN cell number 1, LGN cell number 2, LGN cell number 3, right? Passes back stellate cell number 1, number 2, and number 3 here. Then they start to converge to create this little receptive field that's more like a bar. If we were to draw that over here, what it would look like is that we'd have, we could draw the concentric circles this way. And this is a little, I usually do this on the board. But I'm trying to draw these receptive fields of the LGN cells that happen to be compiled or converge to help create this larger receptive field of the simple cell that we're recording from here. So isn't that kind of cool? We're hitting more of those on-center things here, whereas if you have the bar of light that's hitting it, and let's just draw those same. Area here. I'm messing this up a little bit. If we're hitting it here, we're hitting more of the off-center or off-surround area than on-center. Here we're hitting more on-center. So on, so we get more firing here. Whereas here we have less of that, so we're hitting more of the off region, so there's not much firing there. And then, variations on that theme as we move the orientation of the light there. So this allows our simple cells to be able to actually be within the cortex, to be able to orient and give orientation of bars of light. And so we can actually, they assign these different cells a receptive field accordingly. So again, simple cells being shown here where we have a bar of light. It's hitting the receptive field and making it fire really well. And this is trying to depict how these concentric overlapping receptive fields are being shown here on a patch of retina, on. And as we hit that portion right here, let's go to pen, and we have our bar of light happen to hit right along in here, we're going to get the most amount of firing here. So that's on. And then if we move the bar of light out here, this is more off. It's got to be a faster way to do that. But then that bar of light might be shining over here, and that would correspond to that scenario. So you're hitting off areas here, then you move it over, you hit on here, it makes it fire more, boom. And then you move it over here and it's off again. So this is how this can be depicted. And so what we're looking at is how the this kind of light is being described. So look at this image here and this graph and try to make sure this makes sense to you. I've described some of the major features of it, but kind of work through this and think about it. One thing that they're trying to show here is that, okay, so we're shining light right here along these on areas here. And they're depicting like, okay, the light is off. So we have on center here. And then when they say on and off here, they're talking about, is the light illuminated or not illuminated? So let's just call it that. And so it's when it's illuminated and the light is shining here onto these on center types of cells, then you get some firing that occurs. So so work through that, make sure that you're clear on what on is referring to here, off is referring to here, that this is referring to illuminated versus not illuminated, not the receptive fields, that kind of thing. I think that'll help you be able to see how we're kind of making these different simple cells receiving their receptive inputs. So these simple cells show directional sensitivity. As you move the light across, then you can imagine you're hitting off and on center types of inputs. And so in this layer 4B, where we have some of these simple cells residing, you can have this kind of, they tend to discharge in having more of a left to right, for example, type of movement than right to left. And that kind of organization, you just have to test and see how the cells respond. But there's an actual directional sensitivity, not just orientation, where we're aligning this visual stimulus with the receptive field here. So, okay, and this is another depiction of showing how the visual field here, processed by the LGN and the retina, kind of breaks the stimulus up into little circles here that ultimately get assembled by these cells into being able to create more complicated representations of the cells. And complex cells tend to have a lot more of this directional sensitivity, and hypercomplex cells have even more of a complicated, for lack of a better term, type of receptive field. And we'll go through some of those descriptions here. One thing that's, when we look at these simple cells and how they're actually organized, is and then how the visual cortex is actually organized, is what they found was that as they were recording from the visual cortex, they would track in an electrode and move it along inward and inward, and then ask at each step point, here, 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 and here, whether, what kind of a stimulus would actually cause it to fire. And so that's what we're kind of looking at in this scenario. And so what we're looking at is the orientation of the light here, and how that bar of light is being shown onto the receptive field here. And then each point along here, like let's say we have the electrode track right here, and we measure it, seems to be at this sort of minus 60 degrees orientation, versus if we move ahead, and that's where it has the greatest amount of firing. And then as we move ahead and move our track distance deeper into it, we can find that we find that, let's say this guy right here isn't sensitive to minus 60. It's most sensitive to 60 degrees or so. And so you have these orientation regions here that actually represent columns. And so this is the kind of depiction that we have on the visual cortex. And these represent the orientation columns that we have. So straight up right here, versus slight changes along the orientation of this. And so this kind of columns then have orientation specificity. So here's a column here, here's a column here, etc. And we have input from the right eye and from the left eye that's being segregated at this point. And then what happens is that this visual cortex works in terms of modules. And we have these that are composed of the right eye and the left eye. This represents a small patch of the retina. So what you've done then is that you have part of your retina is being represented and has sensitivity to multiple different orientations of a particular stimulus. Same part of the retina, but you have dedicated processing of multiple orientations to that of the stimulus for that same spot. And then you have from the left eye and from the right eye, and so on. So you have a lot of redundancy in terms of the representation of those areas. So a complex cell is then integrating types of inputs that receive from these simple cells here, and then is able to help create a more complicated type of cortical cell here. So these are composites of these fields here, and contour and contrast are usually what's important here, and we see them in layers 2, 3, 5, and 6. And so as you're moving your visual stimulus across there, you can find these types of changes that can occur within the receptive field, and they're larger and represent larger areas of the retina as well, and have more of a variety of different responses to them. When we're looking at the visual field and the visual cortex, and we take this and we kind of pull this apart. Remember, these different colors here actually, well, you don't remember, but let me tell you that these different colors here actually correspond to different parts of your visual cortex. So your fovea is more out in here, and your peripheral part of your retina is out in this interior portion here. And then this is kind of inputs that we have here. So each small part of your visual field is represented here on your brain. And then if we take that little patch and we look at it, we can see that that little patch has orientation columns in it, cells that are dedicated to processing orientation here. And then we have some color sensitivity regions here, and that we have the eyes represented here. These columns here, the reason we're calling them columns is because there's depth to it. Multiple different layers of the visual cortex that are dedicated to processing stimuli in this one particular type of orientation. So if we look at that and we can kind of see how this stuff is being passed through, there's actually these different types of channels that are responsible for processing visual input. And so what we're looking at in this particular type of depiction here is what we have is a retinal ganglion cell here, like an M-type retinal ganglion cell that then passes the information onto the lateral geniculate nucleus, and then in turn passes it onto the visual cortex. And now we're naming the layers that are responsible for helping to process this type of information. Layer 4C, for example, with sublayers alpha and beta, and then layer 4B being shown here. So these represent the stellate cells that we have that are showing these kinds of inputs that are coming in from the LGN. And then those stellate cells are starting to actually to share their information and integrate them into creating these simple cells that we have residing within layer 4B. So let's take a quick look here at what we have. Retina is here, retinal input coming in through these M-type of ganglion cells that we talked about before, broad diffuse dendritic arborizations. And then what we have are these the magnocellular layers, if you remember, layers 1 and 2 that are receiving inputs from the contralateral and ipsilateral eye. And then these go up to these cortical layers alpha here that have relatively oblong receptive fields here, and then on to these simple receptive fields that I was describing up here in layer 4B. All right. There are this kind of movement depiction what we want to try to remember, and what we can see summarized in the sheet that I'll upload for you guys, is basically trying to show how these different parts of the inputs that are coming in, they represent kind of channels, motion channels here through magnocellular types of input here. And they're processed through distinct types of cells. We have what are known as these blobs, cytochrome oxidase blobs that tend to correspond to the types of cells that we have dedicated to color to the channel processing that we have within here. By cytochrome oxidase, it's a histological marker that's used to help to map the visual cortex. And so when the visual cortex was unmapped, they're just looking for ways of which these cells were actually differentiated from each other and whether that corresponded to any kind of physiology or not. And they happened to find that cytochrome oxidase was a useful one that's indicative of that. And so we can see that here in layers 2 and 3. Layers 4 here is kind of where we have some overlay. And this blue part is representing sort of what's coming in from the ipsilateral eye, and this area here is from the contralateral eye. Or it could be vice versa, but it's one eye versus the other eye, left or right, is kind of being mapped out in this kind of area here. So these tiger striped kind of appearances that we see here. And that corresponds and moves up and down here. But we also have these cytochrome oxidase blobs that are present, all of these multiple layers of the cortex. So for shape processing, then, we have this kind of interesting blob and interblob type of depiction here. So these blobs correspond to cytochrome oxidase positive cells that seem to be organized in these sort of globular-like structures that are in layers 2 and 3 of the visual cortex here. And then there are processes or cells that are sort of in between the two that are, we call them interblob ones. So it was the 70s. They're a little loose with their histological types of characterization of it and how they called it. So everything was blob and interblob is what we have now forever. So retina, input coming in from the parvocellular types of inputs that we have. And so that's more about shape. It has if you remember what those ganglion cell responsivenesses are like for the parvocellular, they tend to they adapt less quickly, but have higher resolution. And so what we look at is that this is feeding into the LGN, layers 4 and 6, or layers 3 and 5. So these represent whether the contralateral or ipsilateral of the LGN. And then these are in turn passed on to layer 4C sublayer beta. So 4C beta feeds in here for processing shape input. Remember, 4C alpha was processing input for motion. So we're already seeing some discrimination in terms of where these things are synapsing in the visual cortex. Those in turn then synapse onto these blob or interblob cells that we have within the layers 2 and 3. So that's what we're looking at in for this shape channel. This is another depiction of how this these large sort of modules within the visual cortex are organized. Input coming in from the left eye or input coming in from the right eye are being depicted in terms of colors of blue and red. And we can see how as we move up these different layers of the visual cortex, how we have orientation sensitivity within a particular column, left eye versus right eye. And then we also have this color channel kind of processing that's occurring here with blob versus interblob cells in layers 2 and 3. And this is depicting how input from the M channel is coming into layers 4C alpha, and input from the parvocellular channel is here. And they call this a parvocellular / interblob channel here in terms of describing this sort of shape input that's occurring here. So this has some color information in it, but also shape. So spend some time with that as you're kind of unpacking that, and make sure that kind of comes for you here. All right, so color, non-M, non-P cells, and also some of it's parvocellular, but we can see this kind of input coming in here from contralateral and ipsilateral retina non-M, non-P cells that are hitting the kind of cellular layers in the ventral to the LGN layers that we have. And that in turn then gets passed on to layers 2 and 3 for helping to, in the blob, to help process color input. All right. So now that we've seen this kind of structure within the different types of layers and the modules that are present within the visual cortex, let's take a peek at what happens in these upper level processing of visual input. And so what happens is we're seeing, okay, retina's coming back, hits the LGN then comes back to the visual cortex then, this is our primary visual cortex that's being shown here, and then the calcarein fissure that's tucked in here. All right, so now what we want to be able to appreciate is that there's multiple different types of visual processing that occurs that corresponds to the channels that we were learning about before: shape, motion, and color. So this kind of input that we're kind of seeing here is shown in this image here. This is actually the LGN kind of cut away, and then what we have is that it's feeding into different parts of the visual cortex, being shown here in the primary visual cortex and right in here. And that gets passed on. So we have a sort of a motion channel here, and then we have that's moving more in the dorsal area of the brain. And then form and color that's being here in the more ventral. And so if we were to kind of look at this kind of mapping happens here, what we're seeing is that the primary visual cortex is then sending this sort of motion processing over to the MT and MST portions, and then the posterior parietal portions here. Whereas if we have this more ventral stream of information, is responsible for processing visual shape and color. So there's some really interesting types of deficits that can occur here. That make it so that you can have a patient who loses sensitivity to motion and loses sensitivity to vision or shape and color, and retains motion. So odd kinds of blindness that can occur in these types of regions here. So you'll want to kind of go through and think about these different types of structures and how they might be related to lesions in the MT, MST, IT regions here, and MST, MT here, and then inferotemporal and the TEO region here, to help be able to, you can link it to these different types of structures or rather deficits that can occur. So have some fun thinking about that part of it. And then this actually kind of gives you some of the answers going through it. So, for example, lesions of the MT or MST can cause this movement agnosia and difficulty in seeing movement. And then in the inferior temporal cortex, which is not about motion, not the magnocellular, but parvocellular type of pathway here, this is sort of processing the what about that. And so they did some really interesting studies of monkeys looking at how they respond to faces and these complex receptive fields that we have. This is a responsiveness to a face here. And they scramble it so that the contrast, percent of contrast remains the same, but it's not organized in the shape of a face, and you just don't have that kind of responsiveness there. So what this is trying to say is, you might have heard people talking about, oh, you have grandmother cells that respond exactly to your grandmother's face in your brain. That's not quite true, but you do have cells that are dedicated to processing stimuli that are organized like a face. And that happens actually, some recent studies show that might even be happening in utero. And so this kind of structure and organization is happening pretty readily. So this is hardwired in a lot of ways, and it helps us to process the what about the image, and allows us to, lesions in this area can lead to what's known as prosopagnosia and a deficit in facial recognition. And so this is the kind of area that can be damaged here. And then in lesion in visual 4, V4 rather, is this visual cortex 4 is where you can see some color agnosia and the inability to perceive color. So this illustrates some of the issues that we have, or some of the types of ways that we've broken down our stimuli. And so we have color, orientation, size, distance, all these types of maps that we have within our visual cortex. And then the interesting thing is, like, well, how do we integrate all of these things together? And this is more of a cognitive type of question that comes out there, and very difficult to do. But our attention seems to help us be able to focus on different aspects of our visual field and do a sort of a detailed analysis of it as it's sort of hitting this broad master map of an image. And that master map of an image, it can vary depending on what we have. But these master maps and evidence of a master map of these images are shown in different types of deficits that can occur. We'll talk about some of those types of deficits in terms of how someone actually processes and thinks about spatial space, even imagining what a space looks like, and that can get damaged. So we'll talk about that in the somatosensation part of it, where we're looking at integration between this visual map that we have here and then a somatosensory map that we have of our body, and how those things can actually relate. And a lot of that's happening in our posterior parietal region of the cortex. Okay, so I know this last part went at a little faster pace. I posted a summary of it so that you can be able to kind of chunk it out as you're going through and spend some time going through it. Hope that was helpful, and I'll see you guys on Thursday.